the clicker.
Yep, cool. Thank you. All right, we're ready to rock and roll. Say, at least half the class isn't here today, right? It's Thursday, so quiz is tomorrow. At least that's what I'm going to tell myself. Hey, why don't I shut the recording off for a second? I'll give you all the answers to the exam, then we'll turn it back on. No, I can't do that. All right, so we're going to continue on with our the subject of genetics, and today we're going to talk about the X-linked and mitochondrial inheritance. And these are the ones that are a little bit unusual, I guess you could say, compared to the autosomal dominant and recessive that we've already seen. So the first thing we're going to talk about very briefly is X-linked dominant. There are no high-yield diseases that are X-linked dominant, with the exception of Fragile X. When I say high-yield, something you're likely to see on a board exam. There are a handful of known diseases that truly are X-linked dominant. Fragile X is considered by many sources to be X-linked dominant, although they're never going to give you a Fragile X pedigree and you're going to be able to identify it as X-linked dominant. And I'm going to explain later on why that is. So the story on Fragile X is coming and the genetics that's associated with it. And one of the issues that I have, and this is why it's really not fair for them to ever test you on a board exam trying to identify an X-linked dominant pedigree, is I don't know about you guys, but I look at that pedigree and the first thing that pops into my mind is that looks like autosomal dominant, right? It really does. Now, in X-linked dominant inheritance, you have twice as many females as males, because basically you only need one of these mutant X chromosomes to show the symptoms, and since females have two, they are twice as likely to have the disease. The problem is, in order to really make a statistically relevant number of females and males, you'd really have to expand this pedigree a great amount. So I would say it would absolutely not be fair for anybody to expect you to be able to differentiate that between autosomal dominant. So unless a board exam is going to give you a very extensive pedigree where you could truly count a statistically relevant number of males and females, then I don't think you're ever going to see that anyway, okay? But it does exist. I wanted to point it out, especially because we're going to be talking about X-linked recessive, and so the natural question is there a dominant? And there is. And later on, I'm going to talk a little bit more about, when we get towards the end of the X-linked section, on some things that confuse X-linked inheritance. I'm going to go over the basics first and just keep things simple, sort of like the model we learned before, but it actually gets a little bit more confusing, and that's where you can actually get some X-linked dominant conditions. One thing I will tell you, though, is very often in literature, in textbooks, all over the place, you'll see people just use X-linked. They won't say recessive or dominant, they just say X-linked. And when they do that, they're always talking about X-linked recessive. If they don't tell you it's recessive, or they don't tell you it's dominant, then they're talking about recessive. The number of X-linked recessive conditions far outnumber any of the dominant conditions. There's only a handful of dominant conditions. And like I said, besides Fragile X, which is absolutely something you would need to know for your boards, but it doesn't follow this pedigree, and I'll explain why, I don't think you're going to see any of the X-linked dominant conditions. There's a couple listed in the genetics textbook if you're interested in seeing them. So therefore, we're going to move on and talk about the X-linked recessive conditions, or once again, sometimes they just call it X-linked. And very frequently, I will just say X-linked. So how do you identify this pedigree? So one thing that you're going to see in the pedigree is it either affects only males or a vast majority of males. Let's say a 10-to-1 ratio, something like that. Very frequently, you'll see in the pedigree it's only males being affected. If the gene frequency is high enough, you may end up seeing some females that are affected, but the males should far outnumber them. So that's one of the things you should look for. So these affected males have unaffected parents. So that looks sort of like autosomal recessive. But one of the things they'll use to distinguish this is there's no male-to-male transmission. So remember earlier when I did the autosomal pedigrees, in each one I said you do have male-to-male transmission. This is where you don't have male-to-male transmission. Okay, so if a male, remember the male has an X chromosome and a Y chromosome. If he has a son, he doesn't pass on that mutant X chromosome. He passes on his Y chromosome. Therefore, he can't pass on the disease to his sons. Although what ends up happening when we do the Punnett square, you'll see that he can only pass on a mutant X chromosome. Therefore, any daughters he have become carriers, right? So once again, males only have one X chromosome. So if we have a recessive condition, because they only have one X chromosome, they're going to show the symptoms of the disease. Whereas females, they have two X chromosomes. So the normal X chromosome would have the normal allele on it. And because the disease is recessive, that normal allele can function to kind of mask the disease allele. Okay, so much, much more common in males. Once again, when you're trying to narrow down a pedigree, really, I think the only pedigree that you could confuse this one with would be autosomal recessive, because the autosomal dominant, you're seeing it in every generation, and as we're going to see with the mitochondrial inheritance, you're going to see it in every generation. So this is the only one where you really see skipping, and that's setting aside the penetrant stuff we talked about before, because that's not fair to test you on a pedigree. So this is the only one where you see generation skipping. So when you see the generation skipping, first thing, if you see, I actually looked first to see if it's X-linked. So I looked to see if there's all males first. If it's all males, I'm like, okay, that looks like X-linked. And then I look, and usually they'll show you somewhere on there, see if you can find the affected males and see if they ever pass it on to a son. If they do, then it can't be X-linked. It's got to be autosomal recessive. Okay? So one of the things I'm going to ask you to be able to do at the very end of my sections, which today is the last lecture, is to be able to identify the pedigrees if you see them. And I'll give you a little thing at the very end of this lecture. We'll talk a little bit about how you do that. Or I'll tell you how I do it, and I also have a figure there that gives you an alternative way of doing it. Some of the real famous X-linked conditions, ones that are high-yield board exam diseases, are Duchenne and Becker muscular dystrophy, and we're going to talk about those today. Lesch-Nyhan syndrome, which, have you guys seen Lesch-Nyhan syndrome? Okay. Usually by the time I give this lecture, I've talked about Lesch-Nyhan syndrome. You'll see that when you talk about nucleotide metabolism, which I think for you guys may actually come in a systems lecture. Very famous and very severe X-linked disease. But you have seen glucose 6-phosphate dehydrogenase deficiency, right? Because I talked to you about that disease. So that's X-linked, very, very common as well. And then hemophilia A and B are also X-linked. So the genes for all of these conditions are on the X chromosome. And we're actually going to talk about hemophilia A and B today as well. So here's our little Punnett squares. And really, in this case, I pulled these figures, by the way, out of a Kaplan review book, which you guys will get later on when you're doing your board review. I believe you'll get the exact book this came out of, maybe an updated version. But in this case, rather than just give you one, they actually gave you pretty much all the pedigrees. I think, or Punnett squares, sorry. I think the only thing they didn't include was if you had an affected female in there, what would end up happening. But the two most common things you're going to see are you have an affected male, what's going to happen, and if you have a carrier female, what's going to happen. And they've already done the crosses for you, so If you have a carrier female, she has a 25% chance of having an affected child. Although if you know the child is a boy, there's a 50-50 chance of her having an affected boy. And if you know the child is a girl, there's a 50-50 chance of her having a girl who is a carrier. And then if you have a affected male, none of his children are going to be affected. Okay, and that's assuming the wife is not a carrier. And so the sons are normal, but both of the daughters are carriers in this instance. We've already done the crosses for you. Once again, with the pedigrees, there's other things I can do. I could give you I could give you a pedigree and say which disease could fit this pedigree, and so you need to identify the pedigree and then be able to say, okay, which one of these is X-linked, for example. Or I could do questions like this. Okay, so what is the genotype of the 3 children on the bottom row on the right? So here we have these 3 children. Okay, so we have an affected father, and we have a mother who's not showing symptoms. So I suppose she could be a carrier, or she could be big X, big X. However, so if we actually go do this, what are the 2 girls? They're not affected, so they're not little x, right? And so they're either big X, or big X, little x. So they have an affected father. He can only pass on the little x, the mutant chromosome, so therefore they have to be carriers. What's the boy? Well, the boy is not showing symptoms, therefore he has to be big X, big Y. There are, by the way, we're not going to talk about them today, but there are Y-linked conditions as well. Very rare, but there are some Y-linked conditions. Okay, so I told you we were going to talk about a couple of the X-linked diseases. So the ones that I chose, just like when we talked about the other pedigrees, there was a couple of diseases I chose to talk about. These are diseases, 1, we haven't seen yet, and 2, there isn't another convenient place in biochemistry to talk about. So for Lesch 9, there's another place to talk about it in biochemistry. So that's why I'm bringing these up. So hemophilia A and B. This is actually, it's written in here somewhere. These are clinically indistinguishable conditions. That means they present the same. So you wouldn't know whether it's hemophilia A and B. And actually, there's a very small percentage of other conditions that can also present the same. There's other clotting or abnormalities, but when you do the math, you'll see that's a small percentage of them that are going to present like hemophilia A and B. So the gene defect in this case, it's factor 8 for hemophilia A and factor 9 for hemophilia B. The types of mutations that are seen in these conditions is a wide variety of possible different mutation types. We didn't even talk about DNA inversions, but you do see deletions, nonsense mutations. So there's not a particular mutation type. You'll notice some genetic diseases like sickle cell, very specific mutation. Achondroplasia was a very specific mutation. Then you have other diseases like beta thalassemia, or these hemophilias where there's a whole bunch of different possible mutation types. That does come into play in the clinic and also when you try to do diagnosis. So because you have different types of possible mutations, the clinical severity varies widely. Okay, and it's based on how much residual activity you're going to have in that clotting factor. Once again, hemophilia A and B are clinically indistinguishable, so you actually have to go in and do some more testing to figure out which factor is defective. Okay, and if you're going to do a very specific factor replacement therapy, which is how you treat these diseases, you need to know which one's deficient. So it's seen in about 1 in 5,000 males. So once again, you don't have to try to memorize the incidence rates, but what I would hope you would be able to do when you see that is say, wow, that's, for a genetic disease, that's really common. For a single gene disorder, that's a very common one. okay? And you'll notice that that's actually more common than myotonic dystrophy, right? We talked about myotonic dystrophy, and I told you it was the most common adult form of muscular dystrophy because generally you don't see the symptoms until adulthood. This one affects children, okay? So this is a much earlier onset, and it's actually more common. Oh, whoops. Sorry. I jumped ahead to the myotonic dystrophy and Becker and Duchenne muscular dystrophy. Sorry about that. It's too early in the morning for me. So forget about what I just said there. Okay. So 1 in 5,000 male. Actually, so the incidence rate is very close to the incidence rate of Duchenne muscular dystrophy, which we will get to in the next section, I promise. But this still should jump out to you as being very common. Okay, sorry about that. Getting way ahead of myself. Okay, so about 80 to 85% of the cases happen to be hemophilia A, which is the factor VIII, and about 14% are hemophilia B. You don't have to try to memorize those percentages. That's just an FYI. You should know factor VIII and factor IX being hemophilia A and B. That's something I think you would be expected to know on the board exam. And once again, there's some other clotting abnormalities that will present the same. And about 60% of the time, the cases are clinically severe. So what do you see in hemophilia A and B? Well, the big issue is you can't clot your blood, and so you get prolonged bleeding episodes, okay? Very often this is first noticed at birth, where you can get prolonged bleeding from the umbilical area, or, and these are males, it's an X-linked condition, so from circumcision. Now, we've already talked about something that might present just like that, didn't we? Maybe a vitamin deficiency? Vitamin K, right? Vitamin K could present that same way, right? Prolonged bleeding somewhere right after birth. So, you know, you'd have to make sure and be able to distinguish between that. Now, with vitamin K, you can just give them an injection. If the bleeding doesn't go away, then it's not a vitamin K deficiency, right? You can get an intracranial hemorrhage during the birthing process, or possibly at any time. So that could be a life-threatening condition in these patients. You get easy bruising, which is shown right here, and this hemarthrosis is also a characteristic symptom. And this is where you actually get bleeding into the joints, which is shown right here. And you could type this into Google Images. I pulled this figure out of your genetics textbook. You could type into Google Images the hemarthrosis and actually get some even more dramatic examples. The most frequent cause of death for these patients is AIDS, which at first glance that might surprise you. It certainly would surprise me, not knowing anything else. Why would the most common cause of death be AIDS? At least it was at one point. And that has to do with the treatment. So you can actually treat hemophilia A and B with factor replacement therapy. So this is where you're actually going to give the person the defective factor so they don't have these prolonged bleeding episodes. And traditionally, these clotting factors were actually purified from human plasma. Okay? So you can actually get human plasma and actually purify these clotting factors out. The problem is this purification scheme. Now, when we say purification, anybody who's worked in a lab, you know when we purify something, we're basically just concentrating it. You never really make it pure. Okay, one problem with this is if the person the serum came from had some type of viral infection, then the virus is still going to remain in that serum, and it's still going to remain after the purification scheme. Okay, and so what ended up happening was thousands of hemophiliacs actually received blood clotting factors that were contaminated with HIV. Okay. It's not just HIV, but HIV was, there's other viruses that certainly can be transmitted in that same manner, but HIV was the one that killed a lot of them. So this was kind of a famous story. It's becoming less and less common because now The blood banks have a much more rigorous screening for HIV-infected plasma, so generally speaking, that's not an issue anymore. However, there's still an issue if you purify clotting factors that way of transmitting other viruses, okay? Including some we probably don't even know about. So a more ideal solution is to do the recombinant clotting factors. And I wrote they're replacing blood-derived factors. I'd have to go back and look. They may have completely replaced by now the blood clotting factors derived from serum. Okay, so you can make the recombinant, and hopefully you understand what I mean by recombinant, right? You clone the gene, you express it in another organism. With these blood clotting factors, they probably undergo some post-translational modification, so you may actually have to make them in cell lines rather than bacteria. I'm not sure about that, but that wouldn't surprise me. But anyway, you can do it in vitro and get rid of trying to use serum. We also have a potential here for gene therapy. If you can give the person a copy of the correct gene, you could potentially cure this with gene therapy. So that's certainly being looked at. Okay, now we're going to get to the story of Duchenne and Becker muscular dystrophy. Just sort of jumped ahead on that last section. So one interesting thing about Duchenne and Becker muscular dystrophy is they are both caused by a defect in the same gene. Okay? And they both actually present the same, although Becker is much less severe. So in both cases, the defective gene is the dystrophin gene, okay, encoding this dystrophin protein. This is a protein that's involved in the cytoskeleton of muscle cells. So we're not going to talk about the function of dystrophin. And you may have already seen the function of it before. Dr. Dan Alishan may have talked to you about it and may even have mentioned this disease. Dystrophin is a huge protein. The protein itself is 427 kilodaltons, but the amount of space it occupies on the chromosome, I forget the exact number, but it's absolutely enormous. And I believe, don't quote me on this, but I believe it is the largest gene in the human genome. Okay, so its enormous size is thought to be contribute to why you see relatively frequent mutations in this gene. Okay, so we saw that also with like NF type 1 was also a very big gene, and you see a high incidence of new mutations in that gene as well. With Duchenne and Becker, so I told you the same gene is mutated. So what is the difference? Both of these are usually caused by insertion or deletion mutations, and the difference between them is with Duchenne muscular dystrophy, the insertions or deletions are usually frame shift mutations of some sort. Okay, so that means the insertion or deletion is not a factor of 3, whereas with Becker, which is actually a rarer condition, you're more likely to have the in-frame insertions or deletions. So if you're just comparing frame shift versus in-frame, you could definitely say, well, the frame shift is a more severe mutation, right? It changes everything downstream, and usually it results in an early stop codon and truncates the protein. So that, not knowing anything else, you could say that the frame shift would be more severe. And that's actually what happens. Duchenne is actually a more severe form of muscular dystrophy than Becker is, even though they're caused by mutations in the same gene. Now, the incidence rate for Duchenne is 1 in 3,500, and that's where I was jumping ahead with, sorry about that, where I jumped ahead. And where I wanted to contrast that with was the myotonic dystrophy, which was the most common adult form. And once again, Duchenne, and actually Becker as well, present much earlier. So Duchenne's going to present pretty early in childhood. Becker a little bit later, but still in childhood. Okay, so Duchenne is actually the most common form of muscular dystrophy. It's also a juvenile or infant, not infant, but child onset. Now you might wonder, okay, well, Becker is less severe, so why is Duchenne much more common? Well, it just really has to do with the mutation type. It's much more common to get, if you're getting the insertions or deletions, it's much more common to get a 1 nucleotide insertion or deletion versus a 3 nucleotide insertion or deletion, okay? So it just has to do with the frequency of the particular type of mutation being able to happen, okay? Here's the clinical picture. This is a picture straight out of your textbook. This kid is actually fairly late in the game. So they're actually normal at birth, and the symptoms start to become apparent by about 5 years of age, okay? They're going to be wheelchair bound by about 10 to 12 years of age and with this progressive muscle wasting, and you can definitely see that. And this kid's probably in his, I'm just guessing, in his early teens. It may even tell you in the textbook how old he is. It does have cognitive impairment, so that, that dystrophin protein does play a role in the brain as well. And usually they die from respiratory insufficiency. Eventually the muscles just weaken to the point where they just don't work anymore. And also the heart muscle is going to get to the point where it doesn't work anymore. I said early 20s. Occasionally, with proper management, you could get a patient that might survive into their 30s with Duchenne. With Becker, basically you're going to have this same progression and the same things, but it's going to happen at a later stage, okay? So the symptoms are less severe, and also they don't become apparent until much later on, okay, about age 11 or so. And they actually live about twice as long. Okay. Once again, this is a good example of a disease to see the effect of a different type of mutation. Okay, and the effect is enough difference where they actually give it different names for the disease. They don't have any current treatment, but Duchenne muscular dystrophy is probably, in my mind anyway, one of the diseases that gene therapy is most aggressively being targeted towards. And Dr. Gardner, I think, is going to talk to you a little bit about gene therapy, and one of the conditions that makes something a good candidate for gene therapy is something that's very severe and there's really no other treatment for it. Okay, because then it becomes less controversial to use gene therapy, as you'll talk about. And this is certainly, and this one's actually pretty common too. So they're really, and they've come up with some pretty neat ways of trying to do gene therapy too, I will say. But in order to talk about those, you really got to get into some of the molecular biology, which we don't have time for. Okay. So I gave you the story of X-linked or X-linked recessive inheritance, and I just kept things real simple at first, and we just thought of things the way we thought of them before. So you have a recessive condition. If you have 2 copies of the gene, and one's normal and one's mutated, well, you're not going to show the symptoms, right? However, now I'm going to complicate things a little bit. Okay, so in females, you actually have 2 copies of the X chromosome, and in males, you have 1 copy. One of the things you're going to learn a little bit later when you look at the chromosomal abnormalities is gene dosage does make a difference. If you have a whole extra chromosome, you're going to have problems. Okay, you know, at the very least, the condition the most compatible with life with an extra chromosome, a non-sex chromosome, will be Down syndrome. Now there are some sex chromosome abnormalities that aren't quite as severe, but the take-home message is that too many genes also cause problems. Now, there are a few particular genes, like we talked with alpha thalassemia, you could have an extra alpha globin gene. That's not really a big deal. You got a whole extra chromosomes worth of genes, though, it causes problems. Okay, So there's a conundrum there. The males only have one X and the females have two. So how do humans deal with that gene dosage issue? Depending on your perspective, and I guess the way I wrote it up was a very male-centric perspective, because I said females have twice as many, so they have too many. You could also say, well, males only have half as many, so they don't have enough. Either way you want to say it, you have to figure out a way of dealing with that. And so what happens is, actually, in females, you're actually going to shut off one of those X chromosomes. Okay? We're going to talk about that process. So that's going to complicate the genetics picture a little bit if you shut off one of the X chromosomes. Because then, well, then females, if you look at that cell and one of the X chromosomes is shut off, then they're really only expressing genes from the other X chromosome, right? Okay, so we're going to explain a little bit more about that. So in females, this process of X inactivation, which is also called lionization, after the woman who discovered this, I think her name was Mary Lyons. This makes most of the genes on one of the X chromosomes unavailable. Basically shuts it off. Okay, it highly condenses that chromosome. Actually, you could even see it under the microscope. That inactive X chromosome can be seen as a highly condensed bar body, a highly condensed chromosome. Okay, and it's actually associated with highly methylated DNA, as we're going to see. So some characteristics of this X inactivation, and this is important to recognize these, because this affects how it's going to affect genetics. The X inactivation occurs randomly. Okay, so which X is shut off is going to be completely random. So sometimes you'll shut off the X chromosome from the mother. Sometimes you'll shut off the X chromosome from the father. Okay? It is fixed in somatic cells. So once that X inactivation occurs, every cell from that lineage is now going to have that same X chromosome shut off. And it occurs, I should have said this earlier, it occurs very early in development. So it doesn't occur at the one cell stage, but it occurs early in development. I think it occurs somewhere around the 100 cell stage, somewhere in that neighborhood. So at that point, the decision is made which X chromosome you're going to shut off, and then every cell derived from that has the same X chromosome shut off. Okay, and because it occurs at random, you end up with females end up being a mosaic of cells in terms of which X chromosome is shut off. Okay. Now that X inactivation is incomplete. What that means is there's a small percentage, about 10% of the genes on the X chromosome that's shut off still are actually active. Okay. So that complicates things a little bit. And when you talk about, for instance, we know if that wasn't true, then you could survive with one X chromosome and be perfectly normal, and we know that's not true. You can survive with one X chromosome, but there are some conditions associated with that, which we'll talk about in the chromosomal abnormalities. Also, that changes the 10% of those genes that are there. It changes the genetics as to whether the gene is located in that section or the other part of the chromosome, at 90% where the gene is being shut off. So how does the mechanism actually work? So there's a region on the X chromosomes called the X inactivation center. And there's a gene within that X inactivation center called EXIST. Okay? And that's only transcribed on the inactive chromosome. So that's part of the region that's still going to be transcribed on that chromosome. And it's only transcribed on the inactive chromosome. They know that because they can look at males who only have one X chromosome and they don't shut it off, and that gene is not transcribed there. Okay, weird thing about this particular gene is that it functions as a long non-coding RNA. This is something I didn't talk to you about, and this is these long non-coding RNAs are actually a relatively recent discovery. And many of the function, like we know in cells, there's a lot of long non-coding RNAs. In the vast majority of cases, we have no idea what they do, if anything. So for many years, they were thought to just be anomalies or junk or something, but now we're learning there's functions. Here is a long non-coding RNA that has a particular function. And what it actually does is this RNA is transcribed and it actually coats the outside of that chromosome, and that begins the process and signals the process to condense that chromosome, to shut it off and to make it inactive. Some of the other things that go on to shut it off and make it inactive are hypermethylation. So when you methylate genes, you shut them off, right? So that chromosome becomes hypermethylated, shuts it off. Histone deacetylation, when you remove acetyl groups from from histones, that makes them more positively charged, therefore they bind tighter. Okay, and then you get further condensation events that end up making this bar body. Okay, so how does this actually affect genetics? Occasionally you can see some female heterozygotes that have one mutant X chromosome and one normal chromosome that are showing symptoms of a disease that's called X-linked recessive. If it used our classical model of dominant and recessive, then that wouldn't make sense. But it makes sense when you understand that some genes are shut off and you have this X inactivation. So if, for example, during the process when the decision is made as to which X chromosome is shut off, just by a random chance, occasionally you will get a female in which most of the X chromosome that's shut off happens to be the normal chromosome, and the mutant X chromosome that contains some mutant gene on it is the one that's being expressed in most of the cells. And then they end up showing symptoms of an X-linked condition. And one example where you can see that is, it's rare, but sometimes you'll get a severe hemophilia phenotype in a female who only inherited one copy of a mutant chromosome because of this extremely unfavorable lionization, where early on in the development, most of the normal X chromosome was shut off and the mutant one remained, and then she showed symptoms of the disease. It also explains why the symptoms can be less severe and more variable in females versus males in conditions where you see this type of phenomenon. Because usually at least some of the cells are still expressing the normal chromosome. Okay, so this really complicates the picture of X-linked inheritance, as you can imagine. Okay, so and one place where this really complicates things is in Fragile X syndrome. This is the example I'm going to use of how this complicates things. So the other examples that we talked about before, they pretty much are just going to follow, straightforward X-linked recessive genetics. Okay, and you don't really have to worry about this. Fragile X, though, is a little bit different. Okay, and this is just a repeat slide. You don't have to. You've already seen this. This is information you should already be familiar with, so I don't have to repeat it with you. But one of the things, we'll look at the incidence. Once again, you don't have to try to memorize the incidence, but that should jump out to you as being common. You get 1 in 1,250 males, 1 in 2,500 females. So it's approximately, you see it approximately twice as often in males as females. Okay? Let's think about that for a second. First of all, that doesn't fit either X-linked dominant or X-linked recessive. If it's X-linked dominant, it should be twice as many females. You should see it happen twice as often in females. Okay? Doesn't really fit X-linked recessive either, because that's a lot of females. Even though there's half as many, that's still, I told you before, with X-linked conditions, it's usually about a 10-to-1 ratio, or even more than that, of excess of males. Okay? So what the heck's going on here? And you know, like I said, one of the things, and I would like you to know that because I'm afraid, even though I believe over time that they're going to reclassify this, I'm afraid you might be asked a question whether Fragile X is X-linked dominant or recessive. In most sources you would look at would call it X-linked dominant. Okay? I'm going to argue that really you shouldn't call it dominant or recessive. But, so is it dominant or recessive? Prevalence in females is less. That argues against it being dominant. But the argument that's placed out there is that it's X-linked dominant with reduced penetrance. Okay, you see reduced penetrance. And we already talked about penetrance. However, what I believe is actually going on, and I don't really think this is even controversial, okay? I think what happens is that there's a certain threshold of cells. So when this process of X inactivation occurs early in the development of a female, it really depends on how much of which X chromosome gets shut off that's going to determine whether or not that female shows the symptoms of Fragile X. I don't know exactly what that threshold is, and the fact that the percentages tell me that the percentage of the prevalence tells me that it's probably somewhere around 50%. I don't know. Or it could be that it depends on a certain lineage of cells, whether that X, in that the cell that that lineage of cells is derived from, whether or not that X chromosome gets shut off. I could also see that happening as well. But it's in all likelihood one of those two things. Does that make sense to you guys? Do you understand what I'm saying there? So whether or not a female is going to show that depends on, you know, how much or what particular cell lineages got which X chromosome shut off. So if a certain percentage of cells or a certain lineage of cells have that mutant chromosome active, then they're going to show the symptoms. So this FMR1 gene, by the way, it is on the section of the chromosome where it's going to be shut off in one of the X chromosomes for the female. Okay? So Fragile X is a good condition to use why the terms dominant and recessive really are not applicable to X-linked diseases. How do you call something dominant or recessive when really dominant or recessive are always assuming both genes are being expressed and one dominates the phenotype? However, in these X-linked conditions, if you have a male, there's only one gene being expressed. And for 90% of the genes in the cells of female, only one gene is being expressed. So I know I went on. It's a pet peeve of mine that they call this X-linked dominant. Hopefully you understand that. But I want you to understand that this does have clinical relevance, and you're gonna see that with the X-linked conditions. All right, I always like to throw this slide up there and ask how many people can't read the numbers in there. Some people don't like to raise their hand. So is there anybody here who can't read those numbers? Wants to raise their hand? I'm sure there is some. There's one. Okay, happens to be a male. Color blindness, at least red-green color blindness, is an X-linked condition, and it's very, very, very common. I think it's about 1 in 10 males. So there's probably a handful of people in here who didn't want to raise their hand. Very, very, very common. And obviously not life-threatening, so they can survive with this. Okay, so the final thing we're going to talk about is mitochondrial inheritance. So mitochondria are the only organelles in humans that contain their own DNA. In plants, you have chloroplasts that contain DNA as well. In each cell, you have hundreds to thousands of copies of this circular piece of mitochondrial DNA. Okay, mtDNA, by the way, is an abbreviation for mitochondrial DNA. But the fact that you have hundreds to thousands is going to confuse the genetics a little bit. Okay. And it's about, you don't have to memorize the exact size, but that's pretty small. That's in the range of plasmids that people use in the lab that they grow in bacteria. It's a little bit on the large side for a plasmid, but certainly I've made some plasmids that are that big before. So that's a pretty small, tiny piece of DNA. So it doesn't encode much. There's 13 proteins on there, and they're labeled. You don't have to memorize them. All of the proteins that are encoded there are proteins in the electron transport chain, but it doesn't have all of the proteins in the electron transport chain. But it's got some of them. But all of the proteins there are for the electron transport chain. It also has a couple ribosomal RNAs and a bunch of transfer RNAs. Those are involved in gene expression of the genes that are on the the mitochondrial RNA, or DNA. Okay, so during fertilization, so one thing I want to mention before we even get on there, because I'm not going to talk about this much beyond this, the vast majority of mitochondrial proteins are actually encoded in the nucleus. So there's a lot of defects in mitochondria, which are actually nuclear genes, and they're inherited in the ways we've already seen. Okay, when we talk about mitochondrial inheritance, we're only talking about the defects that occur on the mitochondrial DNA. Most mitochondrial defects are actually due to nuclear genes. There's far more genes in the nucleus that encode for mitochondrial proteins, okay? So just keep that in mind when we're going through this. During fertilization, sperm don't actually pass mitochondria on, at least under normal circumstances. Very rarely that can happen, but under normal circumstances it doesn't. Therefore, mitochondrial mutations are inherited exclusively maternally. You get your mitochondria from your mom, from your mother. So the mitochondrial DNA you have comes exclusively from your mother. So that plays a role in how things are going to be passed on. Okay, so I think I've told you all the rest of the things on this slide. So the disease is inherited only maternally. So if you have an affected female, all of her children are affected, every one of them. Okay, and if you have an affected male, none of them are affected. Okay, and when you look at the future generations, nobody is affected. So it's always transmitted through the female. Okay, once again, all the children are affected if the female is affected, and none of the children are affected if the male is affected. Okay, pretty simple to remember. These are the high-yield diseases that you may run across, and we're not going to have time to really talk much about them other than to mention them. So I wanted I'm not sure if you cover these in other classes, but these are the three that you'll see in the board review material. So if you're ever tested on a mitochondrial disease, it would always pretty much be one of these three. So Leber hereditary optic neuropathy, which causes the symptoms in the eye. Also, you see a brief as LION. Myoclonic epilepsy with ragged red fiber syndrome, or MRF, and mitochondrial encephalomyopathy and stroke-like episodes. Very hard to say, right? MELAS. So usually you'll see these abbreviated, or you'll see the abbreviations more commonly than you'll see the whole name listed out. Okay, MRF, MELAS, and LION are the three. Okay, and actually, for the most part, the names tell you what the symptoms are going to be. Okay, now, once again, just with the X-linked, when the mitochondrial inheritance isn't as simple as I made it sound. Okay, and this is also where I had to add, I forgot to put something in the PowerPoint I posted, so that's what's in italics here, which I'm going to mention in 1 second. So that's extra. You may want to put it in. I can also upload a updated version with this statement in there. I totally forgot to write it in, and it is important. But because you have hundreds to thousands of copies of this mitochondrial DNA, in most mitochondrial diseases, the mutant DNA is only a subset of the population. And whether or not you have symptoms depends on how much of that mitochondrial DNA is mutant DNA. And so you have to reach a certain threshold before you see symptoms. That also means mitochondrial diseases tend to be hypervariable. They're they have a wide spectrum. Now, don't worry about that with the pedigrees. The pedigree is always going to show you something straightforward. But you can have people who should have the disease who don't, because when the cells divide, the mitochondria, the way they're passed on, you get this random segregation. So just by random chance, some cells might get many mutant mitochondria and some might get none, or just a few, and that affects whether or not you see the symptoms. And that includes in the germ line, okay? So that complicates the picture of the mitochondrial disorders a little bit. And that's called, this mixture and this variable mixture of normal and mutant mitochondrial DNA is called heteroplasmy. So I would like you to know that term, and I forgot to write it in there. Generally speaking, with the mitochondrial diseases, the tissues with a very high demand for ATP are going to be the most affected. So that's why you see very commonly neuropathies and myopathies. You know, muscles, neurons have a high demand for ATP. So the relative demand for ATP suggests whether or not, or the severity of the symptoms you're going to see. And there's no recurrence risk to calculate, right? You know that. Okay. So the last thing I have for you guys, and you could use this table if you want. One of the things I'm going to ask you to be able to do is identify pedigrees. I'm going to tell you the way I do it and the way I suggest for you to do it. You can also follow this. This comes from a USMLE COMLEX review book. This is how I do it. I look for the weird ones first. So what I do is I look for mitochondrial first, because that's an easy one to miss. And actually, based on what I've seen, that seems to be the one the COMLEX always likes to use. But anyway, I look for mitochondrial first. Okay, so I try to eliminate that or identify that first. Okay, if I can cross that one off, the next thing I look for is X-linked, because sometimes people will forget to look for X-linked. So I immediately then start looking. Is it all males in the pedigree? And then beyond there, if it does look like it's all male, start to follow everything else to make sure it looks like it's X-linked. If you can eliminate X-linked, well, now we're down to dominant and recessive, autosomal dominant and recessive. And those ones are probably the easiest ones to distinguish, right? Because the dominant is in every generation, whereas recessive, you see this skipping, okay? And then along with the other rules that go along with dominant and recessive. But if you follow the way that, and you should be able to do them as quick as I just said that, you can identify the pedigrees doing that. Once again, I don't think you're ever going to see an X-linked dominant pedigree tested, because it's too hard to distinguish between that and autosomal dominant. Okay? All right, guys. So that's, I think, all I got for you. And enjoy the next genetics section next week. I'll probably be sitting in on some of those lectures.